in the central computer, in the vasomotor center, signals are generated that travel in the spinal cord, leave the spinal cord, connect in the sympathetic ganglia just outside of the vertebra, and then send neurons that go to innervate the blood vessels, the arteries or the veins. And we had told you that the sympathetic nervous system is truly the main controller, the main instantaneous regulator of the vasculature of the vascular system. And we will see that in a second. Noradrenaline is the major signal used by those neurons. I told you that in most cases the neurons are at the outside of the blood vessels. Only in veins will that penetrate inside the smooth muscle. But in the rule, they're a grid around the blood vessels. And whatever they release has to diffuse to the smooth muscle under those nerve endings. We mentioned to you that the density of innervation varies very widely. You have blood vessels that have no nerves practically, like the very big tubes, the aorta and the vena cava, very few nerves. And then as we get closer to the periphery, the smaller artery, remember those are the faucets to control blood flow and resistance, and there the innervation becomes bigger and bigger. And in the rule, with the exception of the veins, as I just said, the adrenergic nerve innervation doesn't penetrate in the smooth muscle layer. Okay, so let's now try to understand. I told you that those adrenergic nerves have swellings. It's a cable, and at the end of the nerve cell, there are little blobs. 1, 2, 3, 4, 5, 6. And those blobs, which we call the varicosity, this is a varicosity, whenever there is a nerve signal, remember, a few slides ago, originating in the computer, going all the way down, the nerve signal will come to the periphery. And then one after the other, the varicosities will be activated. Because those nerve endings will depolarize. The signal we will, I don't think, oh gosh, so difficult to teach when you have no background, but the nerve impulse will cause a depolarization of this membrane here, and as a consequence, calcium comes in. Always the same thing. Calcium is the activator ion. Whether you talk about nerves, about endocrine glands, about endothelial cells, about smooth muscle cells, about skeletal muscle cells, about heart muscle cells, the signal, the activator ion is always calcium. Calcium is a key signaling molecule for most of our cells, if not all. Okay. And what happens then? Well, before that, the varicosity has been better than you, because you haven't prepared for this lecture. But the varicosity has prepared for the signal. So when the varicosity is resting, it becomes a synthesis place. It takes up tyrosine, an amino acid, from the extracellular fluid. Remember, the extracellular fluid contains everything that the cell needs. So it takes up tyrosine, and a first enzyme will transform tyrosine to dopa, and then the next one to dopamine, and then the final one, dopamine beta hydroxylase. You don't have to remember the names of all those enzymes. Just remember that there are 3 steps. finally the transmitter is formed, noradrenaline or norepinephrine in America. And that is stored in vesicles. And this varicosity is loaded up with such storage vesicles. And when the calcium comes in, the storage vesicles are moved to the periphery of the varicosity, fuse with it, and expel their content. Do you understand that? And thus suddenly, when this nerve is activated, nerve ending is activated, a very large concentration of noradrenaline is created here. And from there it can diffuse to the smooth muscle. So you have the nerves around the smooth muscle. The nerves are activated, they release noradrenaline, and noradrenaline can diffuse to the vascular smooth muscle cells. Everybody understands that? If we ask a question at the test about that, you'll be able to answer? Yes? Okay, good. Now, this vesicle then closes and is reformed and can be recycled. There's a little bit loss of membrane, there's always some loss. But overall, this is a very economical process. You know? It's green. You know? We reuse, like we should do with bottles, like we should do, for 2 generations now, we have thrown away all the bottles. And now they float in the ocean. We have to recycle. Our body recycles all the time. So I repeat, uptake of an amino acid, synthesis to the transmitter, storage in the vesicle. When the nerve ending is activated, there is an increase in calcium, and this makes the vesicle move to the cell membrane, fuse, and spit out the noradrenaline that then can diffuse. Now, we will see in a moment that noradrenaline here can also be taken up again by the nerve ending and restored in vesicles. So at the end of the activation here, there is noradrenaline still floating here, and it's taken up by the nerve ending to be recycled. Nature hates waste, so it recuperates and recycles. Understood? This is a key aspect of nervous function, and it is a very calcium-dependent process, as is shown here, but you don't need to go into the details of that slide. Okay. This slide basically shows you the same thing at the beginning. Tyrosine, dopa, dopamine, noradrenaline here in black, and the release. What happens with noradrenaline once it is released? Well, as I said, it will diffuse to the smooth muscle, and there it will cause contraction. My apologies for the spelling mistake, but I cannot change it because this is a drawn picture. It's not a computer picture, it's scanned, so I cannot correct that. And every time I see it, I feel unhappy about it. So it will activate alpha adrenoceptors, and this will cause contraction of the vascular smooth muscle. So activation of the nerve endings, more noradrenaline, vasoconstriction. Okay? Now, one thing you have to understand when you think about nervous control, the key thing is that the nerve has to keep in control. So the nerve, the degree of activity has to be in full charge of the activity of the innervated organ. With other words, you don't want a nerve cell to give a signal and then the response to go on forever. That's not good. If I tell this muscle to lift my cup and then nothing else happens, I will stay like this all the time. That's not good. So my nerve signals have to continually say lift or keep, relax, keep, relax, keep, relax, and that way I keep under control and I can move around. Do you understand that? It's the same for our blood vessels. We want the sympathetic nerves to tell to the blood vessel contract, but the moment the nerve stops to be active, it has to stop. That's the only way we can maintain a tight control. So the noradrenaline, once it is released, once it has activated the receptors, has to be removed. And this is what we call the disposition of noradrenaline, the disposition of norepinephrine. And what are the possibilities? Well, there basically are three major pathways. One I mentioned already. It is the uptake by the nerve ending. There is a pump here that takes up noradrenaline. And by the way, it will take up also adrenaline, it will also take up serotonin, it will also take up dopamine. This pump will take up any monoamine, but let's stick to noradrenaline here, and put it back inside the nerve terminal where it can be restored, reused for release. Okay? This is a very powerful mechanism, but interestingly, it only functions, and this is something that we discovered a long time ago before you were born, it only functions in between action potentials. So, when this action potential comes in here, this pump is switched off, as long as the membrane is depolarized. So if you generate a high concentration of norepinephrine, you block this so that you don't release and take up at the same time. You release or you take up. Okay? So very simple, so to speak, but of course at the cellular level quite complex. So this is a very powerful pump, and it's important not only for adrenergic nerves, but for many nerves. And for example, one way to block the pump is to give cocaine. Have you heard of cocaine? You know that this is a very powerful drug, and I wish none of you ever has used it. But cocaine works by blocking this pump, the same cellular mechanism, and in the brain that has the consequences and leads to drug addiction, etc. So it's a very important mechanism, but it is used by all neurons, that are involved in the release of either dopamine or adrenaline or noradrenaline. So that's one way. The other way is simply that it is washed away. Remember, our extracellular fluid is washed continuously by the changes. What causes the washing of the tissues? Who remembers? That was the equilibrium between two forces. You remember? Who knows? Come on, kids. Don't disappoint me. Remember hydrostatic pressure, colloid osmotic pressure? That was causing a washing of the tissues all the time. Well, part of that will take noradrenaline with it, and it will appear in the circulation. And a third uptake is by the end organ itself, by the smooth muscle, that takes it up and then destroys it to non-active components. So once the activity of the neuron stops, noradrenaline is removed very rapidly. And this permits to keep control of the situation. I mentioned, to be complete, that where I speak only about noradrenaline or norepinephrine, that certain adrenergic nerves can contain other transmitters as well, but I think at the level of this course, it's not important that you remember which. Only remember that in certain blood vessels, other things come to complicate the picture. So, what do we need to remember? Maybe not the first paragraph, but the neurotransmitter, once it is released, is removed from the junctional cleft, that's the space between the nerve ending. This we call the junction, and the cleft is the space between the neuron and the smooth muscle. Between the junctional cleft, in between action potential, through neuronal uptake and extraneuronal degradation. And in certain cases, other transmitters can be released. This is another schematic that basically shows you the same thing with a little bit of color and in smaller print. I wanted to show it to you simply to make the point again that the noradrenaline, once it is released, will activate cell membrane receptors, and depending on the tissue, the pharmacological properties of those receptors can vary. We can have alpha receptors, subtype 1, subtype 2, and then further different subtypes. We can have beta adrenoceptors, 3 different subtypes, and I've shown you that table already, I believe. If you're interested, you can look at the different possibilities. But for us, for this particular course, in terms of vascular control, you have to remember alpha 1. That's the one that is important for the control of vascular tone. Oops. Good. So, we have a sympathetic nerve that is activated. And let's assume that the nerve is continuously activated. So it generates one action potential after the other. So it will release a continuous amount of norepinephrine. The next topic that I would like to discuss and share with you is the concept that, of course, the central computer generates impulses that go to the periphery, that tend to maintain the blood vessel vasoconstricted, but can local factors affect it? With other words, at each moment in time, is the amount of noradrenaline released determined only by the activity of the neurons? And the answer to that is no. It can be modulated. And the reason why I want to share that with you is because many years ago, and we spend a lot of time demonstrating that, and I think that this has become an important part of our thinking about vascular control. And let me show you how we came to that. Many, many years ago, I worked in America, and at that time I was using isolated blood vessels, veins, And I was using electrical stimulation. I've already shown you slides with electrical stimulation. And here I would switch on my stimulator, and I knew that I was stimulating nerve endings, and I then kept it going. And you know, normally you would expect a continuous contraction as long as the electrical stimulation is going. At this point here, I injected acetylcholine. And just to show you how powerful this type of response is, you see that it's immediately switched off the relaxation. And when I removed acetylcholine, it came back. To me, that was very, very puzzling. Particularly when I started to look and forget about the two top traces, I want your attention only for this middle panel. This is an experiment done on a tibial artery, not on a vein. It doesn't matter what artery, on an artery, and it doesn't even matter that it is an artery on another blood vessel. And I did two things. I used two concentrations of acetylcholine during electrical stimulation, and here, remember, electrical stimulation stimulates the adrenergic nerves, causes action potential in the adrenergic nerve varicosities, and this will cause a release of noradrenaline. Okay? That's what I just explained. I hope you still remember that. Otherwise, we're in deep trouble here. So we have a constant activation of this calcium-dependent release of noradrenaline. So in this experiment, I give noradrenaline. I inject noradrenaline. And you see, I have a sustained contraction, and now acetylcholine does nothing. Well, here it causes a dose-dependent relaxation. Who can explain that? How could that be? So, the two experiments. This experiment, I switch on a stimulator that activates nerve endings to release a constant amount of noradrenaline. Here, I give a constant amount of noradrenaline. Here nothing happens. Here we have a full relaxation of the preparation. And this is the same as what I showed you here. See? So how can we explain that? Let's go back a few slides. So, this nerve ending, when the electrical impulses come, calcium will go in, noradrenaline will be released, and that will cause a contraction. In the second experiment, I'm giving noradrenaline. So here the concentration of noradrenaline is determined by me. Acetylcholine does nothing. So what do you conclude from that experiment? Where does acetylcholine work in the first case? Where does it work? On the smooth muscle or on the nerve ending? Nerve ending. It works on the nerve ending. It is working here, not here. Because if I give noradrenaline, nothing happens. If the nerves are activated, it switches off the release of noradrenaline. And I went on to measure that. There are tricks to measure the release of transmitters. I went on to measure that, and this would be during nerve stimulation, the release of noradrenaline, and if I give acetylcholine, it causes inhibition of that release reversibly. And this then explains this response. This is a very powerful response, very rapid. You know, in this system, acetylcholine is as potent as tetrodotoxin. Do you know what tetrodotoxin is? Have you heard of tetrodotoxin? The puffer fish? You know, the fish, I don't know how you say it in Korean or in Chinese, but this is the fish that the Japanese love so much, you know. But you need a very well-trained person to prepare it, because if the person doesn't prepare it well, if there is still toxin, you die, okay? You've heard the stories. That's the puffer fish, tetrodotoxin. And tetrodotoxin is one of the most violent poisons that we know, because it stops nerves from working. Well, in blood vessels, acetylcholine is as powerful as tetrodotoxin to switch off the release of noradrenaline. Now, this was my first experiment. It was a very important one, because it gave me a PhD degree, or the equivalent, and ultimately that's why I became professor at the University of Antwerp, because I did those experiments. So to me those are very important experiments. But they were only the beginning. I will not show you all the experiments. Over the years we were able to find, this is the receptor that I started off with, that I just showed you. That there are many other receptors. Histamine. Remember histamine? Where did we talk of histamine? Who remembers that? When we talked about capillary exchanges, histamine was a substance that was released when you scratch yourself or when the mosquito bites you, okay? And remember, it would open the arteries, increase capillary permeability, shut down the veins. Well, one of the reasons why it opens the precapillary vessels is because it inhibits, it disconnects the vessel from sympathetic control. Okay? Then there are others that do it. Noradrenaline itself, you know, noradrenaline release can, if there is a very high concentration of noradrenaline, it will feed back. You know? So if we go to that situation here, if noradrenaline, if the nerve is activated, becomes more and more active, the concentration of noradrenaline will increase and increase. Above a certain level, it will feed back on the nerve ending and say, hey, enough. We don't need more. That's what we call a feedback control, where the transmitter itself feeds back when it reaches a certain concentration and tells the neuron, you've done enough. You don't need to release more. You understand that? So there are a number of things that can inhibit adrenergic transmission. Among those, I will spend a few moments on that, the local metabolites, potassium, pH, acidosis, Professor Dangmei, osmolarity, all of those will cut down the release of noradrenaline. But there also are factors that will augment it. And the one that you want to remember is angiotensin. Remember, we talked about angiotensin. If there is angiotensin around, this will be a very strong stimulus for all nerve endings, for all sympathetic nerve endings, to release more noradrenaline. And I think I told you, and we will come back to that, that angiotensin 2 activates the sympathetic system considerably. And this is one of the things that it does. But let's spend a second about this here. When our skeletal muscle contract, I have already told you that, there will be changes in the extracellular fluid. There will be a greater concentration of hydrogen ions. There will be release of breakdown products of ATP. The potassium ion concentration will increase. The osmolarity will increase. All of that will cause the vascular smooth muscle to relax. And remember, this is something that plays a role to adjust the blood flow to the needs of the tissue. So if my tissue is active, the products of that activity will open up the faucets so that more blood can come. Well, at the same time, those products will all work to inhibit adrenergic neurotransmission. And thus, in working muscles, the muscles become automatically disconnected from the sympathetic system, at least as long as we're not in extreme situation. And this is good, because as we will see, if I exercise and suddenly my blood vessels open up, the peripheral resistance will decrease. And if you remember from the first lecture that I gave you, pressure equals resistance times flow. If the resistance decreases, if the pressure remains the same, there will be more flow. But if the resistance decreases too much, we will have to do something to maintain the pressure. We will have to cause constriction. Well, that constriction will not happen in the working tissues because of that. So the computer can say, okay, let me increase, we will see that a little bit later, let me increase sympathetic outflow to everywhere, and in function of the degree of activity of the tissues, they will contribute or not. So tissues that are not active will be maximally compensating for the tissues that are active. And again, think a little bit about that, because it makes the system once more very efficient. We call that functional sympatholysis, which means the fact that those nerves are cut off when the tissue becomes more active. Again, try to understand that, because that's an important concept. Always the same thing. We have to keep the whole organism going, but if the muscle, if I'm running to save my life, because a lion is running behind me, at that time it becomes very important to keep blood going to my muscle. Just one example. And we can go on like that and give you many of those. So this brings us to the concept of, I just talked about the concept of pre-junctional. Now in skeletal muscle or in the brain, we would call that presynaptic. But what we discuss, the structure of the nerve endings around the smooth muscle is not a real synapse. It's a junction, so pre-junctional inhibition. And there are several aspects here that I mentioned and that you may remember. One, the pre-junctional adrenoceptors prevent exaggerated release of neurotransmitter. Two, the pre-junctional inhibitory effect of metabolites ensures functional sympatholysis in the working tissues. And I mentioned histamine, that the pre-junctional effect that will allow the local inflammatory response that we mentioned in a previous lecture. Okay? You understand that more or less? Or less or more? Try to do the best you can. Again, if you don't ask questions, I accept that you accept and that you understand. If you don't understand, you have to ask me questions, okay? So, Don't say later it's too complicated. No, it's not too complicated. It's too complicated because you didn't ask questions. Okay? Good. Next one is the parasympathetic nervous system or the cholinergic vagal system. Now, the vagal system is very important for cardiovascular control, but not for the vasculature. It is one of the key systems that regulates the frequency and the contraction of the heart. Because there is a yin and yang between the adrenergic nerves and the cholinergic nerves. And I'm sure that if you have lectures on the function of the heart, Professor Peter Kang, I would imagine, or maybe Professor Dong Mei Wu, will tell you about cholinergic control of heart rate, for example. That is very important. But in the vasculature, it's very limited. So I will not spend a great deal of time on it. There are a few blood vessels that are innervated by cholinergic nerves in the gastrointestinal system and in the genital organs. But most of the tissues don't seem to have it. We don't see the nerves. Now, if there are nerves, they're just regular cholinergic nerves. Now, it's the same story as what I told you for noradrenaline. There are nerve endings, the action potential comes, calcium gets into the nerve ending, the nerve ending is prepared, has synthesized acetylcholine that is stored in vesicle, calcium comes in, the acetylcholine is released, and then there is a reuptake and a resynthesis mechanism, exactly the same thing. Nature has no imagination. Don't forget that. It uses the same tricks over and over again. And then acetylcholine will go to the other side and cause activation of a cholinergic receptor. Again, depending on the tissues and the species, other substances can be released at the same time. Is this really important? To be honest, after 50 years in research, I'm still not convinced that in you and me, cholinergic nerves are important for the control of vascular tone. Yet, experiments like this have been performed by my master, by my professor. When I was your age, I was working with a very famous professor in America. And he did this experiment, and he took a student like you, and he measured the blood flow in the forearm. And then, at one time, the student didn't see what was going on. He said, "Oh my God, you're losing all of your blood." And immediately there was a very big increase in blood flow. And if he gave atropine to block acetylcholine, he did not see that. So Professor Shepard was convinced from that simple experiment that cholinergic nerves were very important. But it has been very difficult to demonstrate them. So we still don't know, after all those years, how important it is. And I still believe that when we talk about blood vessels, and nerves, we have to focus on adrenergic nerves. And if there are cholinergic nerves, I think they are important because of this. With other words, that when cholinergic nerves are activated in the blood vessel wall, if they release acetylcholine, rather than acting on the smooth muscle, they will mainly act to switch off the release of noradrenaline. We will in the next lectures talk about the endothelium and acetylcholine, and although this is a very powerful mechanism, I still believe that this work, this action, my PhD thesis, in terms of physiology, is the most important action of acetylcholine in the blood vessel wall. But I'm biased about it, of course, because those were very exciting experiments a long, long time ago. Okay? So, remember, there are certain areas, that are innervated by cholinergic neurons, but the exact contribution is uncertain. Gastrointestinal system and the genitals, maybe. And it is likely that cholinergic vasodilator innervation results mainly in prejunctional inhibition of the release of noradrenaline. Now, besides parasympathetic nerves and cholinergic nerves, are there other nerves? Well, yes. There are nerves that produce nitric oxide and nerves that produce peptides. Now, actually, if you remember some of the slides that I already showed you, of those co-transmitters, as we call them, here you see that substance P, and in the one that I showed you before, you see peptides again, So there are peptides that can be released by adrenergic or cholinergic nerves, but there also are nerves that specifically release peptides at their ending. Also, sometimes sensory nerves are in the tissues and they will release peptides and they can contribute to vascular control. But that's an area where I think Professor Dongmei Wu knows the stuff much better than I, and I will let her decide whether you need to hear about that in other classes. There is only one other non-adrenergic, non-cholinergic nervous system. This is another, for years we talked about that. Now you understand when we talk, okay, we knew about adrenergic nerves, we knew about cholinergic nerves, and then we put all the others in a bag that we call non-adrenergic, non-cholinergic nervous system.
What's the difference between neurotransmitters and neurotransmitters? What's the difference between neurotransmitters and neurotransmitters?
Well, a neurotransmitter is any substance that is released by a nerve ending. A neuropeptide is a peptidergic neurotransmitter. So it's focused. Neurotransmitter would be noradrenaline, catecholamines, noradrenaline, dopamine, adrenaline in certain cases. Would be serotonin, that would be in that class, monoamines. Or would be acetylcholine, that's another one, or glutamate, or peptides. And the peptides would be substance P, would be CGRP, I believe in certain cases, would be intestinal, the vasoactive peptide. So there are a number of peptides. And now the neuropeptides are a subclass of the neurotransmitters. Okay, good. I'm glad. So obviously I didn't explain it, so you asked. That's right. So the ones that I will say, just show you one slide about, are the nitrogen nerves, because we will spend a great deal of time talking about nitric oxide when we talk about the endothelium. And nitrogen nerves are nerve endings that will transform, as we will see, L-arginine into nitric oxide, because they contain an enzyme called neuronal nitric oxide synthase. And this then is used as a transmitter. And it will, in the innervated organ, cause a relaxation, as we will see. We'll come back to that. You will understand this part of the slide much better later, because it activates the production of cyclic GMP. And this system is inhibitory and antagonizes excitatory signals like the adrenergic nerves. Now, this is a fundamentally important system in erectile tissues. So in the corpora cavernosa of males, there is no erection possible almost without the production of nitric oxide. And I'm sure you've heard, have you heard about Viagra? Yeah? Everybody has heard about Viagra. Well, Viagra does nothing else but protect this and the cyclic GMP, that normally is broken down very rapidly. It will protect cyclic GMP from breakdown. So it makes this system much more efficacious. And that's why Viagra is so good, for the treatment of erectile dysfunction. But again, illustrating one aspect of nitrogen pharmacology that I will not spend too much time on. But I would like you to remember it, that in certain tissues, in particular in erectile tissues, that other nerves contribute, and that nitrogen nerves are among the most important. And those are fundamentally important nerves for the species. Because if there is no erection, there is no sexual intercourse, and thus everything stops, the race stops. So important nerves, but not for everyday's, well, for most people at least, not for of importance every day of life. Well, the adrenergic nervous system remains the main controller. And those non-adrenergic, non-cholinergic nerves are all of local importance. They're very localized in certain parts of the system. Now, it's amusing because, as I was about to say, we talked for many years of non-adrenergic, non-cholinergic nervous system, which is a very sophisticated name, and we abbreviated that into NANC nerves. And if you go back to old papers of about 15 or 20 years ago, you will see NANC nerves. And you may wonder what NANC means. Well, NANC means this. With other words, means I don't know what I'm talking about. But always the same. I will give you other examples of that. When scientists don't know what they talk about, they invent a nice name and they immediately abbreviate it. And then they sound, very scientific, but for many, many years we talked about these bloody things and we didn't know really what we were talking about. And it's only more recently that those two groups of nerves have emerged. Okay? We have to keep the milieu intérieur, the internal milieu, satisfactory for all cells of the body. I told you that this was achieved by the circulation. I gave you the basic rules of the circulation, and we now have been through the different components and the different size of blood vessels. Now, somehow, all of what I told you has to be orchestrated, coordinated, has to be able to react to moment. And this is where the reflexes come in. Do you know what a reflex is? A reflex is something that you do not control, that comes automatically. And that can be in the motor system, or that can be in the autonomic system. The motor system, the one that you know best, is the one of the doctor, tapping on your knee, and then you extend that's a reflex. I mean, there's no way you can control that, Well, it's the same for our daily life inside our body. We have a number of reflexes. We will talk about the ones of importance for the vasculature, but there are many other reflexes. All our bodily functions are controlled by reflexes. When we have to go to the toilet, when we, the way we breathe, all of that is reflexly controlled, always to make sure that the totality of the system functions properly. Okay? Now there are a number of major reflexes and control systems that contribute to our well-being, and in this lecture, second lecture today, I would like to go over those. And the first one I would like to discuss are the arterial baroreceptors. Remember, the basic formula was pressure equals, what was it again? Who remember? Flows times resistance, pressure. So, and remember, we have the faucets. And what did we need to adjust? So the faucets would be open or closed, but the water tower had to remain at a good level. So we have to keep pressure at a constant level, or eventually increase it if needed, we'll see that. And then, if the pressure is maintained constant, then in our different tissues the faucets can open and close, and we will always have enough blood going through all the tissues. Okay? We understand that. So this implies that something senses it. You know, if you have a pump, you're engineers, if you have a pump system with an automatic system, there is a sensor to measure pressure. Now, so that if your pump generates too much pressure, you cut down. It's the same principle. Now, the sensors that are responsible for that, we call the baroreceptors. Barometer, you know that word. Pressure measurement, you know. Barometric, you know. Baroreceptors. And those baroreceptors are located in two areas. There are nerve endings that will sense pressure in the blood vessel. With other words, what could they measure, do you think? To what would those receptors be sensitive? If the pressure goes up, what will happen to the blood vessel? Stretch. So they're stretch receptors. They will sense the amount of tension, the wall tension in the blood vessel. If the pressure goes up, the wall tension will augment. They say, oh, if the pressure goes down, the wall tension will go down, and the sensors will sense that. Again, talking to people with bioengineering background, that at least should not be difficult to understand. And those stretch receptors are located in two areas. Just in the aorta, just as the blood comes out of the heart. That's the general feeding line for the whole body. And there is another specialized area, and you probably don't know this, but the aorta goes here, and then it has two branches that go to the brain, the carotid arteries. And in the carotid arteries, then later on, splits into two branches, the external and the internal carotid arteries. And the external will give the blood here, to the outside of your skull. And the internal is the main pathway to feed the brain, to feed the black box, to feed the computer. And at that level, there is a swelling of the carotid artery internal, the branch that goes to the brain, and that is loaded up with those sensors. So continuously, we measure the pressure of the blood that goes to the brain. You remember that I told you already several times that in the order of priorities, number one is the brain. Because we can live 20 minutes, 30 minutes, 1 hour without blood to our hand. We can cut it off. We can bring it back to the hospital. We can, the surgeon can do his thing and it will eventually recover. But the brain 7 seconds, 8 seconds, without blood, finished. So it's very crucial that we continuously monitor what goes to the brain. And those sensors then send their impulses to the cardiovascular sensor, and these will regulate the amount of sympathetic drive that goes to the heart, Professor Peter Kang, or the amount of sympathetic tone that goes to the spinal cord, to the sympathetic ganglia, through the veins and the arterioles, through the release of noradrenaline, of course. Okay? And this is the same picture from the textbook, I think, that you use, or that you should use. I think that comes from there to show you that. Now, I just want you to know the existence of those systems. I don't want you to understand how they function, because that's way beyond yours and probably my level of understanding, so I will not insist on that. But what you may want to know is that if we look at nerve activity, that means those nerves here, if the receptors are stimulated, they will activate, there will be a degree of activity in those nerves. Well, if we look at that and we measure that in function of the pressure, in the aorta or in the carotid sinus, you see that the carotid sinus nerve will become active at lower pressure. It's more sensitive. And again, this is very logical because the carotid nerves will determine how much blood goes to the brain. So it's very sensitive. Now the rest of the body is sensitive too, but not as crucial. This slide is probably the best one that I show you to illustrate this order of priority. The fact that above all, at each moment in time, whether you're awake or asleep during my lecture, your brain has to receive blood, and that one of the ways to monitor that is by measuring the pressure. And thus, if we take those carotid arteries and we occlude them, which you can do by pressure, by pushing here on the carotid arteries, or through surgical procedures, this, if we occlude those, this creates an enormous reaction. Here in an animal, I was occluding the carotid arteries, and you see, whoop, blood pressure went through the roof. Because suddenly, a major supply of blood to the brain was interrupted. And that's alarm, 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 alarm. And if the blood doesn't go through the artery anymore, how could more blood go there? Well, that means that the resistance is too high, the flow is zero, pressure equals resistance to flow. If flow drops down, the resistance goes up and the pressure goes up to try to overcome the occlusion. And several blood vessels participate to that, but we have no time to go. I have time, but I don't want to go into details. Another thing I'd like you to remember about those pressure receptors relates to a disease called hypertension. You know what hypertension is? High blood pressure. Very common. I'm sure if you talk to if all the fathers of the students in the room, probably 50% of them have too high a blood pressure. And I take drugs. I take 3 drugs every day because my blood pressure is too high. Yeah? Now again, to understand blood pressure, you have to assume that something goes wrong with the baroreceptors. And indeed, if this would be the normal response of the baroreceptors, so as activity, as the pressure goes up, the response you see that in hypertensive animals and humans, the whole thing is shifted to the right. So, if I don't take drugs, my computer feels happy at a much higher blood pressure. Now, we don't know why that is. And we do something about it to keep it down, because we know that too long exposure to high blood pressure on the long run is bad. But all people with high blood pressure will tell you that. They actually feel better when they're off the drugs. But it's not good for their blood vessel long term, so we keep the blood pressure down. But I feel more active, more ready. So we don't understand why this is, but we know that it is part, that we cannot have chronic hypertension if this shift does not occur. So our sensors are deregulated, and this allows then the blood pressure to be higher. I don't want you to remember the details, I want you to remember the principles. The repressor receptors, the baroreceptors are there to keep perfusion going, mainly to the brain. If something goes wrong with that, we can become and have a chronic high blood pressure. Okay. So, in a sense, the carotid sinus and the aortic baroreceptors will ensure that at each moment in time enough blood goes to the black box. We all agree on that? But is that enough? The body has another sensor, because you may have enough blood, but if the quality of the blood is not good, then you run into trouble again. So we have what we call the chemoreceptors. And those chemoreceptors, this is what I just told you. This is the internal carotid artery. That is a main source of blood to the brain. There is another artery that brings blood to part of the brain. It's the occipital artery. And at the beginning of that occipital artery, also side branch of the carotid artery, there is a swelling. And that swelling contains these babies, sensors, chemical detectors, cells that are sensitive to changes in pH, in oxygen concentration. They will measure the quality of the blood. And mainly they will measure, is there enough oxygen? Is there not too much CO2? Not acidic? So that the quality of the blood is continuously monitored. Again, how they do that, that's a different story. I want you to remember that there is a quality control of the blood going to the brain at each heartbeat, continuously. Oh, I did something wrong. Okay, so how does that function? Well, these experiments here, or this schematic, shows you what happens with vascular resistance in function of the PO2 of the blood. If there is 100% saturation of the blood, then everything is under control. If the PO2 goes down, progressively the resistance goes down. The blood vessel open up. We try to compensate. And with too much CO2, we have the opposite. If we have too much CO2, we want to get more blood to the brain. So this mechanism implies that if we have a poor composition of the blood at the level of the chemoreceptors, too little oxygen, too much CO2, the blood pressure will go up. And thus more per time of the bad blood will go to the brain. But you know, if you have blood that, let's say, has 80% good quality, if you send more of that, ultimately the total oxygen supply will be sufficient. Do you understand that? If I have, let's say, blood that contains 100% oxygen, I need, let's say, 1 liter per minute to the brain to give enough oxygen to the brain. Now suddenly my oxygen content goes down. Well, if I send more blood to the brain, the total oxygen delivery will still remain good. I still give priority to my brain. I still give priority to the black box, to the computer. So this is what you have to remember. Chemical analysis, signal, make sure that we get enough oxygen to the brain. If the blood is lesser quality, let's send more. But make sure that we give enough foodstuffs and oxygen to the brain. Okay. So those are two very important. The next one I want to say a few words about are the low pressure mechanoreceptors. And those are basically stretch receptors again. The same thing as the carotid sinus. But they're located here, in the atria mainly, and in the large veins. And what they measure, remember that I told you that the large veins contain 75% of the blood volume. So those receptors here, remember all the blood comes back here to the heart and then can be pumped through the lungs, etc. These receptors continuously measure the filling of the system. Again, if you have engineering background, you know that for a pump to function properly, it has to be primed. The liquid has to come to the pump. And if there is not enough liquid coming, the pump cannot function properly. Do you understand that? Now, these receptors measure the filling. So if blood volume goes down, for example, if we bleed, these receptors will automatically be activated. And something will be done. And the something that will be done will be, an increase in blood pressure in different things, exactly the same as a carotid sinus response. If our pump isn't filled properly, we are in danger. We have to do something. And those receptors activate that. And at the same time, those receptors will activate the hypothalamus, and we've talked about vasopressin, an antidiuretic hormone. And these are the peripheral receptors that will help to control that. As a consequence, this will determine they will help to determine not only blood pressure, but also the amount of urine that we release, that we form by secreting antidiuretic hormone. And thus, if we are too full, they will sense it, and we will produce less antidiuretic hormone. So we will pee more. If we are too empty, they will produce more antidiuretic hormone, and we will pee less. Now you all know that. You all have experienced that. If you jump in a pool and you float around, hang around in the water, after a while, what do you have to do? You have to go out of the pool and go to the toilet. Why is that? Well, that's because when you jump in the water, suddenly the gravity changes. And blood that was trapped in your legs is now moving to here. And thus you stretch those babies. And those babies tell, hey, 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 we're too full. Let's stop producing antidiuretic hormone. And then you have to go to the toilet. Very simple. Basic mechanisms to control, to make sure that the pump keeps turning properly. 70 times or more per minute. Every minute of your life. There has to be enough blood filling the heart so that it can work properly. And those are major contributors to that regulation. It's a fascinating field. Again, all you need to know is that those things exist and what they basically do. Ergoreceptor. I will not talk about respiration because that will be covered in another field, but certainly you will understand, I hope so, you will understand that, for example, the chemoreceptors will have an impact on respiration. If the chemoreceptors sense that there is not enough oxygen or too much CO2, we will try to breathe in more to get more oxygen in. But there also are specific reflexes related to respiratory movements, but I will not talk about those. I want to say a few words about ergoreceptors in particular and proprioceptors in general. You have any idea what proprioceptors are? Proprio means yourself, means knowledge of what you are. No. You all know that. If I close my eyes here and I do this, I know exactly where my hand is. So our computer continuously informs us of where we are in space. And it does so because there are stretch receptors everywhere. In the joints, in the muscles, I know whether my muscle is tense or not. Okay? I know that because of proprioception. But that's conscious. I can do something about it. But the same receptors feed into the cardiovascular centers. The receptors in skeletal muscle will feed into the cardiovascular center. And they will affect sympathetic traffic. And I think I may have time at the end of the lecture to say a few words about exercise, but let's do it right now, because that's important. Let's assume that you're in a starting block, ready to go, waiting for, I don't know whether you run marathons or any form of track work, but you have to start working or walking or running or whatever. Your brain knows that. The brain, before you start moving, the brain will tell the vasomotor center, do something about it, and you already prepare. Your heart starts beating faster, and you get, less sympathetic tone in the muscles that will have to work, etc. We call that central command. Then, besides that, of course, during exercise, if I start exercising, my blood vessels will dilate because of the metabolites, and this, of course, will decrease peripheral resistance, will tend to reduce blood pressure, and thus my presoreceptors will kick in, my baroreceptors will kick, the carotid arteries will kick in, and because I use more oxygen and I produce more CO2, my chemoreceptors will kick in. So it's a very complex thing. But beyond that, there also are receptors in skeletal muscle that are sensitive. And when we contract the muscles. I know it, you know, but my cardiovascular system knows it too. And if this happens, they will tend to increase sympathetic tone to the blood vessels. And now there is a very big difference, and that's what I want you to remember. I think I may have said something like that already. Very big difference on the type of exercise that you do. If I simply walk or run, we call that isotonic exercise, you know? Because basically I move the legs, but the tone in the muscle doesn't really change a lot. Now this will create the reaction that I just mentioned, central command, chemoreceptors, baroreceptors, and a steady increase in blood pressure with the degree of work. Nothing to be alarmed for. But if I do isometric exercise, you know, weightlifting, and I hold there, or in nature, hanging from a branch above the ravine, you see, life-threatening, very important. There my muscles clamp down. Isotonic exercise. And as you may, isometric exercise. And as you may remember, during isometric exercise, what is the big difference between those two? Is that here, my muscles can be fed in between contractions, but during isometric exercise, the muscles are so stiff that they compress the blood vessels, and no blood can get through. Okay? Now, I know black box is essential in all of that, but here I am, hanging from that branch. If I let go, I drop. Potentially I'm dead. Thus it is absolutely important under such conditions to get enough blood to my working muscles. And the only way we can do that, think of my carotid artery being occluded, it's the same thing. My blood vessels in the skeletal muscles are occluded. The only way to get over that is to increase pressure. Bioengineering, just an engineering problem. The resistance is bigger. The only way to get fluid through it, I mean, it's like blowing in a balloon. It's always the same thing. You need to increase pressure. And blood pressure goes skyrocketing to maintain that. Now, this is potentially a dangerous situation. What will happen with the brain during that period, do you think? You remember what we talked about? What would the brain circulation do? Who remembers? Nobody? Don't disappoint me. Remember what happens with the brain circulation as blood pressure goes up? There is an area that we called autoregulation. So, for most of those changes, the circulation will auto-regulate. So the brain itself will not be exposed to those extreme blood pressure. Remember this slide? As the blood pressure increases, on the one that I just showed you, up to here nothing will happen. Brain is protected by autoregulation. Now, if the blood pressure goes higher still, then it breaks through, and then we get into problems. Or if the brain circulation is too fragile, then we get into problems. Okay? We break through and we get into trouble, and that's why isometric exercise is potentially much more dangerous for health than isotonic exercise. And why you have to be careful with that if you don't want to have problems. So, isotonic exercise you cannot do wrong. Isometric exercise you can do wrong, if your blood vessels are fragile. Remember that. And if your boyfriend wants to have big muscles, tell him to be careful, especially later on in life when his blood vessels become more fragile. The last major regulation that I want to spend a few minutes on is thermoregulation. Thermometer, you know what it is? What do we measure with a thermometer? Temperature. Good. At least I got one second answer that I got today, so I'm happy. Now, thermoregulation is extremely important. And of all the regulations that I just mentioned, if I had to list the one that is ultimately predominant, it's thermoregulation. We have to keep our body temperature at 37 degrees centigrade. Why is that? Well, because that's the optimal temperature for most enzymatic reactions in our body. And as you know or may not know, but we are a big chemical machine. We continuously transform things chemically, biochemically. And most of our enzymes operate at an optimal temperature of 37 degrees. Very simple. And thus, our body goes at great length to ensure that temperature remains constant. And one thing it does, there is in the hypothalamus a group of neurons that are specifically sensitive to temperature. They are thermometers. They continuously monitor temperature. And they adjust in consequence. And they adjust the cardiovascular flow, the sympathetic flow, they adjust that to the skin. I think I told you already that in our species, the skin is the major site for thermoregulation. So if we get too warm, too hot, our blood vessels dilate. We turn pink. If we get too cold, our skin blood vessels constrict. We become pale. And that is normally controlled by the hypothalamic center. But of course, it's a little bit more complicated because there also is a peripheral perception that will feed into the central system. And if we need to vasodilate, this will happen at the levels of the arteries, that will happen at the levels of the vein, and we will also have secretion of bradykinin, I mentioned that will help to open up so that we exchange more heat, or that we can sweat more, that we can release more sweat. So it's a very complex system again, but primarily determined by the centers in the brain. And when something goes wrong with that, we get fever. Fever is a resetting of this center. For some reason, and I don't have to go why, but somehow those centers, by the presence of bacterial toxins in the blood, for example, those centers are fooled, and they are reset at a higher level. And thus we have fever. Okay? Let's keep that. Of course, remember, we also have what I just described is the central thermostat. But we also have that in each room we have a thermostat or a radiator. And remember, that was the direct effect of the temperature on the blood vessels. And that I already have explained. I don't have to come back to that. Remember that when we cool, we get a constriction, when we warm, we get a relaxation, and this is because cold is making the alpha receptors on the smooth muscle more sensitive. Okay. Now, let me very rapidly go through. I think I've given you the major, very brief outline, the major regulation system. One concept that I'd like you to have in mind is that not all circulations are equal under the sun, and that depending on the circulation, they contribute more or less to those different reflexes. Okay? For example, if we go at the brain, well, the brain is not, we don't really know how well or if the brain blood vessels are innervated. The brain blood vessels are pretty independent. Their major regulations are metabolism of the brain cells and autoregulation. Autoregulation, vasodilator metabolism. Neuronal, we don't really know. I personally don't think it's very important. You want the brain blood vessel just to care about the well-being of the neurons. And it's up to the rest of the body to make sure that I can do that. So brain, not much now. What about the coronaries? Well, theoretically, the coronaries have acetylcholine, or, the heart is innervated both by cholinergic and adrenergic nerves, and the coronary arteries contain some alpha adrenoceptors, but ultimately, those play very little role again. The major thing for the coronaries are the vasodilator metabolites in the heart itself, and then beta adrenoceptors that are sensitive to catecholamines, to noradrenaline and adrenaline in the blood. Those are the major players. If we start running, remember our adrenal gland will start releasing adrenaline and noradrenaline, and immediately our heart will kick in. Okay, so these innervations will mainly be of importance to control the function of the heart muscle, and the rhythm, the rate at which it beats. Blood vessels will follow. The kidneys, very much dominated by the sympathetic nerve system. There are other areas of importance. Autoregulation is important. Prostaglandins, we haven't talked about those, but they're important too. But this is a system that is truly very much dominated by the sympathetic nervous system and the hormones. Vasopressin, angiotensin II, aldosterone, etc. So every time we have an emergency situation, our kidney shuts down. We can wait to cleanse the blood. We can wait to pee. That's not life. And the kidney has, receives about 20 or 25% of cardiac output, so it's a major source of saving blood for other purposes, if necessary. Okay? So kidney, major contributor to vascular reflex regulation. Skeletal muscle, well, remember Professor Shepard and the student being told that he is bleeding. This slide, we made that up with Professor Shepard, so I had to put this one in. I'm not certain it's true. There is innovation, but remember that this will be interrupted when the muscle becomes active. Remember at the beginning of the lecture, pre-junctional inhibition, functional sympatholysis. Again, vasodilator metabolites are crucial. But in resting skeletal muscle, there we have a big reserve for vasoconstriction if necessary. Because don't forget, or I don't know whether you know that, but skeletal muscle is of course the biggest amount of active tissue in our body. Well, for some of us it's fat, But you know, for young skinny people like you, it's skeletal muscle that is the major active tissue. So if we are resting and we need to do something, you know, we can switch off circulation to skeletal muscle to save more blood to go to the black box. The skin, well, a little bit the same. But here we come into the big problem that the skin blood vessels are dominated by temperature, and that this both at the central level and at the local level. But looking at their contribution again, the skin is one of those territories where the body can shut off blood flow if necessary. And in many reactions, if we lose blood, how will the doctor know that you are possibly losing blood? Because you turn very pale. You shut off the skin. And if your thermoregulation is okay, that's not a problem for a prolonged period of time. The splanchnic circulation, well, the splanchnic circulation is another one that is a major contributor to reflex regulation. Because the function of the splanchnic circulation is, if we eat, we have to digest. And then we have to take up what we digest and bring it to the liver. That can wait. That can wait. We can interrupt that process and save the blood that goes to the splanchnic, to the gut, etc., if we need it for something else. So splanchnic circulation contributes majorly to all the major reflexes. So skeletal muscle, skin, and mainly splanchnic circulation and kidney. Those are the players in those reflex regulations. And of course the veins we've already discussed. I already explained to you that in terms of the regulation, it are the splanchnic veins that are located around the center of gravity that will be the regulators of the filling pressure of the heart. And that will keep those low pressure baroreceptors happy. The veins. Okay? So, what you have to remember is that the contribution of the different vascular beds to the reflex regulation of the circulation varies. The cerebral and coronary circulation are favored at the expense of the other ones. We want the computer to work and the pump to pump, at all costs, because if we stop that, we're dead. The others, we can wait. As I say, we can wait to digest our meal. We can wait to go to the toilet. Not always, but we can. You know, those are not crucial things for life. So we can switch off the kidney. If we're in thermoregulatory control, we can turn pale. You know, skin can live for hours without temperature, without blood. That's not the problem. Okay, very rapidly, integrated responses. Of course, in the second lecture I've taken one example at a time, one mechanism at a time. Now in our daily life, that's not the way it goes. You know, we do many things at the same time. For example, if we look at, we won't talk about diving, although diving is the most extraordinary cardiovascular thing. When you dive in the water, you know, amazing. But most of us don't dive every day, so I will not go into that. Emotion. Emotion is an incredible thing because it involves the cerebral cortex, and then it goes to all the things, and emotion can contribute, can invoke reactions, vascular reactions in almost all the organs and systems. And time? Can I have 2 more minutes? Yeah. You can look at those sides, but you know, emotion, and that's individual. Some of you, if I aggress you by asking you a question, you will turn red, the other will turn pale, you see. Some of us, when we're emotional, Our heart beats faster, not all. Some of us sweat, some don't, etc. So you can look at that. The same for exercise, but exercise, thank God, I already discussed. So here you have it: central command, thermoregulation kicks in, of course, because as we exercise, we generate more heat. So all of that comes in, and you can find that there. The posture is the one where I spent a few, because it allows me to show you my favorite slide once more. Posture. We're standing here. I'm standing here. I'm starting to bleed in my legs, because those valves, remember, open up and blood. And that's okay. We can cope with that. My pressure receptor, my low pressure, this will be sensed here. Less blood coming back, so we have all kind of reflex adjustment to posture, and we can measure that. But the reason why I want to show you that is because posture is probably the best example where we can show the predominance of thermoregulation. As long as I stand still here, I can stand for a long time, except if I become too hot. If my temperature goes up, now I'm standing, and automatically, reflexly, my skin vessels will close down, my kidney will close down, everything will close down to keep, enough blood going to the heart and to the pump and to the brain. But let's assume that I'm in the sun and I'm very warm. Then my blood temperature will go up. And what will be the consequence? Well, you all know. If I stand still and I'm heating up, thermoregulation will dominate. And thermoregulation will say, I have to open up the cutaneous veins to exchange heat. I have to open up the cutaneous circulation to lose heat. And the moment I do that, suddenly the blood supply to the heart is not sufficient anymore. The heart cannot pump. No blood to the brain. I lose consciousness and I fall down on the ground. This slide, I show you that. You don't have to know why, but to illustrate that in this system there is a preponderance. So, I'm done with my lectures on homeostasis and vascular control. I hope I have convinced you that this is a rather intriguing and complex system. Ultimately, this serves to maintain, to keep the milieu intérieur under livable conditions, and please remember this order of priority. But also remember that in the regulation, things interact and interfere, and that there again there is an order of priority. I should have here one more paragraph saying, in terms of reflex regulation, bang, 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 this is the order of priority. I have to add that on for next week. But altogether, I think our cardiovascular system achieves the mission that I discussed at the beginning. That is to make sure that the cells of our body, particularly those of the brain, are continuously bathed in solution that contains all that they need and in which they can excrete their waste products. I want to mention that my understanding of this topic comes from my years of work with John Shepherd, with whom I had the privilege of writing 2 books. He passed away a few months ago, and when he died, I felt that I had lost my father for the second time. So, thank you very much, and I'll see you in a week or so.